for coming back. Sorry to cut your break short, but I think we've got a really good session here. I'm gonna, I guess, introduce myself. I'm Graham Marsh, Scientific Director of PSTC, and I'm sure you've seen emails from me leading up to this meeting. And thank you all for joining. I'm gonna lead off the session with a brief talk about a recap of our meeting last year, some takeaways and lessons learned, and then we have a really nice discussion planned about standardization and qualification work streams that are ongoing globally. And so you can see the list of designers here. We'll introduce them as we go, but I think that this is going to be a really interesting session about how we see these models being adopted globally. So just to give you a recap. So I think Klaus was supposed to have this picture in his slides, but you can see it now for the first time, about CPATH being a pre-competitive neutral ground. And so that was a really good opportunity for us to serve as a conduit for the first workshop for this discussion, looking at how we can bring together industry, model developers, academia for interfacing with regulatory agencies. And the first meeting was a good baby step in terms of initiating some of these qualification discussions. And I'm glad to see that this is growing and continuing. The first workshop, you have all the materials in the pre-read from the meeting invitation, but again, here's the QR code with all the slides and materials from the first workshop. We kicked it off with a talk from Jeff Siegel, who works with York at in Otis, talking about the qualification pathway. And this has not changed in a year, so York explained this much better than I could. But we looked at ways that we might qualify liver complex in vitro models as tools for assessing drug-induced liver injury. And we talked about how to include them either as part of a weight of evidence submission, sort of ad hoc, or going through the entire iStand program. And again, so this is where we brought together, scientists from the FDA, model developers, academics, and pharma scientists for this iterative discussion around how we see applications of these models and what are some realistic context of uses in the DILI space that we could pursue for qualification. Speaker lists, some familiar faces in the audience again. But because we were focused so much on liver, we talked specifically about DILI applications. We brought in some clinicians. Mark Avigan from the FDA and Eric Cohen from AbbVie are both clinical hepatologists who gave us a good insight about how the field sees applications for these systems for assessing DILI. And then we dove into context of use, as Sonia said, very important, and then looked at some of the analytical and biological performance considerations that would need to exist or be qualified or be validated to qualify a model for those context of uses that we discussed in the first session. And then on the second day, we had Tina Morrison from the FDA and Skip Hayashi, who's another clinician at the FDA, talk about credibility assessment frameworks and DILI assessment. And so we're going to lean on some of Tina's materials for the breakout discussions tomorrow in terms of how we think about developing context of uses for specific unmet need or questions of interest. And so for the scope of this project, again, because we're looking at regulatory applications, we ignored is probably the wrong word, but we didn't look at applications where these models would be used for hit ID, lead optimization, target screening, and really dove into what are the evidentiary considerations that we need to have for the regulatory applications looking at either efficacy or toxicology to show safety in an IND or an NDA package. And so this is all liver focused. And so when we think about analytical validation considerations for the liver, there's, a lot of them, but from a baseline level, you want to make sure that your liver chip is producing albumin and urea at a level that is commensurate with what you'd see in vivo. And then because we're looking at drug-induced liver injury, we want to make sure that the liver chip is producing the right enzymes, the right transporters that it would have an appropriate response to a drug added as part of a DILI assessment. And then the biological performance considerations, and this is all laid out in much more detail in the white paper and the slides from the meeting, but as you think about translating your in vitro model to a clinical assessment, what biomarkers or what endpoints are you going to validate to show that there's translatability and the model is predictive? And so there's a lot of choices. ALT and AST are obvious because those are the clinical endpoints that people think about for assessing liver health in a human. But there's also GLDH or albumin production or urea or cytokine release, all of which are easily accessible on the liver chip because it's an in vitro platform. But you might have to do a little bit more legwork to show translatability to the clinical outcomes that you're seeing. And so for the breakouts, because we were focused on liver, we had 4 groups, and they were context of uses that were, we'll say, from realistic to aspirational going down the list. And so the first is using a CIVM as a tool for stratification of patients in a clinical trial. So how would you use patient cells to predict where there might be a hepatic injury in a vulnerable patient group, and help that inform your patient selection for a clinical trial. De-risking a drug for known in-class toxicity. That is very similar to what York spoke about with the emulate context of use that was just developed in iStand. Complex feature models is a tool for retrospective analysis. And so we talk a lot about using the systems as predictive, replacing animals preclinically, but they're also potentially better tools for clinicians looking to invest a mechanism of DILI that's observed in a clinical trial, and would probably be more predictive than an animal or other 2D in vitro assessment that you would be done after the drug is already in humans. And then the last is using complex in vitro models as a tool for immune-mediated DILI. So there are not good tools, just full stop, not good tools for immune DILI right now. The animal models and the current in vitro tools are not predictive. And so what sort of qualification would, or what sort of development would have to occur for qualification of an immune-mediated DILI model? Yes, so the successful regulatory applications for a complex feature model would have to, one, fill a gap or unmet regulatory need. That is the clearest, starting point is what need are you fulfilling with the complex feature model? Have a clear use case, and then a well-defined context of use to enable this qualification. And so, as an example, coming out of the breakouts, potential context of use statement would be complex feature model is a tool for assessing idiosyncratic DILI observed in a clinical trial of a small molecule. And the use case would be there that if you saw elevated ALT or AST in a clinical trial, but it doesn't reach a Hy's law threshold that kills the compound, could you use a complex feature model as part of a weight of evidence assessment to continue using this molecule if you can understand the mechanism of DILI better in this 3D tissue model? And then that specific COU will drive the analytical validation considerations and the biological performance considerations that are necessary for validating and qualifying the system. And so we are all here in this room right now, and this is the circle that we're stuck in right now, I feel like, and I've shown this at a couple of presentations. And so regulators, whenever I speak to somebody in a regulatory body, they want more data to be comfortable making decisions about using complex in vitro models for regulatory decisions. Developers are looking for guidance from the regulators, and that guidance can't come until the regulators have more data. And the models that developers make inform or enable pharma to test their drugs and submit those data to the FDA. And so this cycle is something that these discussions can help us break out of. These face-to-face or virtual face-to-face interactions are critical for finding context of uses and applications of these systems where we can break out of this cycle and actually move the field forward. And so with that, thank you. This is, I think, similar slide to what Nick started with, thanking, but this is just the people that were helping out with that first workshop. And again, QR code that I think you already have in your invitation email to this meeting.